Okay, thank you, Christina, for the invitation, and I'm very happy and excited to be here presenting Pathogenex, the RNA atlas of human bacterial pathogens that we have launched very recently. And I'm gonna talk about what we have done with these data and what we have been doing, and also hopefully could convince you that this could be this is a really good resource for machine learning applications to answer complex questions in infection biology, and of course our plans on applying machine learning on these data. But before I start to talk about that, I would like to mention how it started, why we needed such a database. It just started with like when I had a project that we wanted to see how a pathogen that is called Yersinia tuberculosis can survive for a long time in a mouse model, for which we call persistent infection. So there we wanted to compare the expression profile of early stages of infection and late stages of infection. And we ended up with 1,200 or more than 1,200 genes that are differentially expressed when you compare those two conditions. And then, of course, the first thing I've done was to look at what kind of genes are they and what kind of picture it can provide to us. And then it was the functional class chain of the genes it kept. I mean, that gave quite important information to us to understand what really is going on. But this was just based on like 41% of these 1,200 genes. And the rest, we had no any idea or any annotations on them that would give some hints. And it's, I mean, it's worth to note that there were like 195 genes that were annotated as hypothetical proteins that we don't really know nothing about them. And by that time, I was like looking for a resource that I could compare those genes that would any transcriptomics data that would give me some more hints. And then, of course, there was not so much of that. And then was like to look at the function of the genes. Then, I mean, GO, the gene ontology database, is one of the largest database that you could get information from, where they try to predict the function for genes to annotate, and that is based on direct experimental results or sequence comparisons. And it's good to note that that there are not really actually many genes in GEO. Also, when you look at the total number of protein sequences in UniProt, so there are 15 million protein sequences in UniProt, and only 0.3% of them are experimentally characterized. And here in this list on the right, you see the organisms that have more than 1,000 genes annotated. And as you can see, these are mostly including well-studied organisms and only a couple of prokaryotes in them. So not so much information on them. And then I was thinking like a condition, some kind of experimental setup that I could put my strain on it, and then that would mimic host conditions, that is stressful host environments, such as like low pH or nitrosative stress or oxidative stress, low iron, low oxygen. And if I could put my bacteria on those conditions, and then maybe I could get some information on those genes, what kind of responses they are giving. So those will be then as I mentioned just earlier, some in vivo mimicking conditions together with a control, and then a virulence-inducing condition. A virulence-inducing condition is not described for, of course, many pathogens, but it is described for Yersinia tuberculosis, where we see that the virulence arsenal of Yersinia tuberculosis are induced. And then, of course, this was for Yersinia only, and you can imagine that this will be very costly and time consuming also. But of course, if you would include more bacterial species in this list, such as bacteria from different gram staining, with different oxygen dependency or extracellular, intracellular lifestyle, that would put more significance on the comparisons when you do. And that's how idea came. And then of course we could do this by when I saw the RNA tag sequencing that allows up to 52 libraries in one tube, which reduces the cost to 90%. Of course, it reduces the biasness also in the library preparations. So that made it possible. So then we started to gather strains from different labs that are expert in growing and in of course the pathogens. So I visited the labs and we designed an experimental setup like the stress conditions that would be applicable to most of those strains, which we try to keep the stress conditions the same as much as we can for the accuracy of the analysis that we're gonna do later on. And it's good to note that we kept the growth medium and the growth temperature optimal for each strains, but the stress exposure almost the same for all. So we could then get the differential, and we can get the expression profile, and of course the differential expression for over 100,000 genes. And we saw that the bacteria are very actively and dynamically respond to changes in the environment by regulating their transcriptome from 62 to 90% in at least one of the stress conditions. And not a surprise that the highest fraction of the regulated genes were found under hypoxia, nutritional downshift, and stationary phase, where the environment is quite significantly different than the control, and the exposure time in those strains on those conditions were higher. And then of course it's to do a cross microbial comparisons, then we had to know which genes are the same from different species. And then of course there are ways of doing it, and we choose to go for functional orthologs with the KEGG, which class assign a KO number for genes that have a similar function independent of sequence homology, and then also for isofunctional homologs, which have a degree of sequence homology and the function. As you can see here, the gene groups that are clustered together are larger in numbers in KEGG, but it might contain not exactly the same genes. And it's opposite for the isofunctional homologs. So you get a smaller number, but more similar genes together. But we kept those two groups of genes, and then it was time to kind of find out the probability of a gene group to be differentially expressed in a certain stress condition. So we to assign that score, we took we looked at the proportion of the regulated genes in each group, and we looked at the frequency of dysregulation among all species we have, the 32 species. And then we looked at the number of the regulated genes in the group. And that ended up with an equation that you see here. And we compared this equation with a well-described test or statistical test, such as a hypergeometric test that is used for gene enrichment studies, in which we saw that these performed much better than that one. And then, of course, is then to look if this PATHOgenex score could really predict the probability of the genes to be regulated in certain conditions. So what we have done, we then ranked the genes with highest PATHOgenex score in each conditions, and then we looked what are those genes that appeared on the top. For example, here I show for low iron, and then you see the genes that have very high PTDX score in low iron are actually genes that are involved in iron acquisition, iron metabolism, and the same for oxidative stress too. So, and then when you look at the PTDX score for KEGs and the PGFAM, the isofunctional homologs, you see that we see the same genes mostly, and you see that even though the size of the gene groups are higher in the KO groups, we have almost similar level of PTDX score in isofunctional homologs. Then we thought that is mostly due to that the more similar genes gather together in the isofunctional homologs, so they get a similar PTDX score. And then we decided to continue with the PGFAM groups. And PTDX score also is actually a transformation of many, differential expression of many genes from all the species we have in the database to a single value, which then simplifies the cross-microbial comparisons or the gene regulation in a comprehensive way. So now we see that HMPA gene from different pathogens are mostly regulated in nitrosated stress. And then, of course, we could do more with this one, and then we went to look at how the stress responses are similar in bacteria that we tested. And then, as you can imagine, that the gene content and phylogenetical order of the gram-negative and gram-positive are different. Then we separated the gram-positive strains and the gram-negative strains, and then we recalculated the PTDX score for them, and then we looked at how the stress responses are similar in different pathogens. In different groups of pathogens. And as you can see here, it's a very known fact that the response to low iron oxidative stress and nitrosated stress could actually overlap, and that we see in such exercises also. And you can see that this is quite different in gram-positives. So once we have looked at these similarities, then we could go and see what kind of biological pathways or processes are employed in different stress conditions, or if there are some that are overlaps. And now here you can see the PTDX scores of genes that have similar patterns over hypoxia, nutritional downshift, and stationary phase could be involved in the biological processes that you can see here. And then the same for low iron nitrosated stress and oxidative stress. So this is an example that from gram negatives, and we have done this for gram positives too. And then the question is like, what kind of genes would be more, or potentially be important for bacteria to stay or to maintain infection to maintain in the tissue or the infection side. And then we were like, maybe the genes that are responding to many stress conditions could be important for the adaptation of bacteria in the infection side. And then we looked at the gene groups that have high PTDX scores in at least 6 of those stress conditions. And we looked for the conserved genes that are conserved in gram negatives and positives also. And we found, and we call those genes inverse stress responders. And we could identify 168 of them that are involved in basic biological processes such as metabolism, protein processing, RNA processing. And interestingly, we found 9 of those to be known targets for antibiotics, which indicates that the others in this group could be a potential target for novel antibiotics. And also the genes that have similar expression patterns to these USRs, when you go and look for expression patterns, could be maybe potential targets for novel antimicrobials. And I must say that we have looked at also species-specific USRs, so which means that we look for genes that are uniquely found in one of the species in these 32 species in the database and responding to multiple conditions. So these could be also an important target for like precision medicine, for example, for a certain type of infections. And then we could see, we were like, I mean, are these responses really active during infection? So we looked for the data that we could get from the literature and found in vivo data from two clinically important pathogens, Pseudomonas aeruginosa and Staphylococcus aureus. And then we compared the responses that we found in our database. And then we saw that that 75% to 83% of these responses that we could detect in at least one of the stress conditions that we tested in vitro were actually responding during real infection scenario. And we found some VSRs also that are responding to these stresses in vivo. And more interestingly, actually, we found some genes that the regulation does not match to any regulation that we found in all these 11 stress conditions that we tested, which indicates that there's an in vivo specific regulation for those genes. And all these data is already published very recently, and it could be taken from the website, in an interactive website called Pathogenex.org, where you can look at for the regulation of operons. For example, here I put the type 6 secretion 1 operon in Pseudomonas aeruginosa. You can see that it could be induced in oxidative stress, acidic stress, and inhibited in stationary phase hypoxia. You can look for the gene groups, how the gene groups are regulated in different stress conditions, and then you can go, I mean, directly from the gene groups to the genes, exactly the gene, I mean, itself, from different species, how they are regulated under those conditions. And of course, you can get the expression levels also. So this is what we have done so far, and now we are also still working on it, and of course, much more can be done with this data.